You were once one cell big. How did you come to be several thousand trillion cells big? In a nutshell, the answer is by cell division. And I think that whoever sent this set of text messages, though their spelling is a little wanting, did a pretty good job of summarizing it. Basically, you start with one cell which grows in size and then pinches in half until it forms two smaller daughter cells that are identical to the parent cell. There are many reasons we should care about cell division. If it occurs too fast or not enough, some very problematic things can happen. If cell division happens too quickly, then a tumor can form, such as formed in the salivary gland of this gentleman. Luckily, this tumor was harmless or benign, because after having it removed, he was able to survive for another 30 years without problem. He was lucky that the tumor did not spread anywhere other than the salivary gland beneath his tongue. Not everyone is so lucky, particularly those that get cancer of the large intestine or colon. Sometimes when a tumor forms on the inside of your large intestine, like right here, you can see a bunch of cells that are dividing way too quickly. If they continue to divide, they might break through the surface of the large intestine and start moving inward until eventually they reach a blood vessel. And that's really bad news, because then the cancer cells can jump into the blood vessel, swim through the blood vessel until they get to a new tissue, some new organ elsewhere, and then start to crawl out of those blood vessels into the new tissue and form a new tumor in a new area. This process is called metastasis. And when metastasis occurs, this abnormal kind of growth gets a new name. It's called cancer. Colon cancer is bad news, because not only will tumors form in your colon or large intestine, but those cells might metastasize into these blood vessels, take the blood vessels up to other organs, most often the liver. The liver has many important functions: detoxifying our blood, creating bile to neutralize stomach acid, and so on. So as you can imagine, once those cancer cells get up here and start forming their own metastases, or tiny tumors that take over the liver, the patient is no longer going to be able to survive because they won't have any working liver cells, just a big mass of cancer cells instead. So when the cell cycle is not properly regulated, cell division can occur too fast, leading to the formation of a tumor, which can lead to cancer if metastasis occurs. Yikes. So then, why do cells divide at all? Why not just exist like this amoeba, growing by engulfing a small little food item right here using some pseudopods, and then slowly let your cells get larger and larger until you turn into a huge amorphous blob and just exist that way? Then you have no cell division and no cancer, right? Sounds great. Well, there's a problem with that. As cells get larger, they get less efficient. Imagine one smaller cell right here, and then another cell that's twice as big, and then another cell that's 3 times as big as the first one. You can see why large cells are much less efficient than small cells. First of all, the nucleus in really large cells is much more stressed than that in small cells. This nucleus is going to have a lot of trouble trying to send enough messages out of the cell and into the cytoplasm as is needed to boss around the entire cell. While this cell will have an easier time getting its messages out, because it has less messages to send. And this cell is going to have a very easy time, because it only needs to send out a few messages to control the very tiny cell. So the tiny cell is more efficient because the nucleus doesn't need to do as much work. Furthermore, this cell is going to have a lot of trouble getting as much food as it needs, while this cell is going to have an easier time. This is best understood by looking at the surface area to volume ratio in cells. Surface area is the outside of a shape, like what I'm coloring in on this cell right here. The cell membrane makes up the surface area, and as you know, cell membranes control what goes in and out of cells. If a cell wants food, it needs to use its surface area to let the food in. Meanwhile, the volume is the total space inside the cell. The volume determines how much food the cell needs. To survive, a cell needs a high enough surface area to volume ratio that it can get enough food to feed itself. As a cell gets larger and larger, Yeah, the surface area does increase. Like here mathematically you can see it increase from 6 centimeters squared to 24 centimeters squared to 55 centimeters squared. That's great. So it can get more food over time. But the volume is increasing even faster. It's going from 1 centimeter cubed all the way up to 8 centimeters cubed, jumping all the way to 27 centimeters cubed. The volume is increasing faster than the surface area is. It starts off with 6 times more surface area than volume, then suddenly it's only 3 times larger, then it's only 2 times larger, and if you kept going like this, eventually the volume would get way bigger than the surface area would be, and the cell would not be able to meet its internal demands. Therefore, being a cell with a large surface area to volume ratio, more surface area than volume, is a good thing. So cells tend to stay small. So never fear, the world is not going to end by the destructive force of the blob, an enormous cell that tries to engulf everything, because its surface area would never be large enough to feed the vast volume inside it. To avoid inefficiencies like this, cells divide. Cell division can be simple in some cases and harder in others. It's particularly easy if you are a prokaryote. As you guys remember, pro means before, karyo means nucleus, so prokaryotes don't have a nucleus. Instead, they just have a single wound-up circular chromosome of DNA depicted right here that can then be copied, moved to opposite sides of the cell, and then the cell wall and cell membrane can grow down and in until eventually it divides into 2 cells, and voila, prokaryotic cell division is completed. It is called, therefore, binary fission, binary meaning 2 for the 2 cells, and fission meaning splitting in half. And one thing that's very distinct about binary fission is that the 2 daughter cells are completely genetically identical to the mother cell that started off up here. Though eukaryotes also have a phase of growth, a phase of copying their DNA, and then a phase of dividing, the eukaryotic cell cycle is indeed much more complicated. It starts off with this first cell growth phase known as G1, then moves into the DNA replication stage. To replicate means to copy. This stage is also known as S phase. And the S stands for synthesis, or making, of DNA. So it copies its DNA, and then it goes into its final phase of growth, G2, in which the cell grows even larger, and starts to move organelles to either side of the cell, preparing for division. And then we've got the division itself. Often this part of the cell cycle is simply called cell division. Sometimes it's also called M phase, because during M phase, the cell needs to go through the bother of dividing its nucleus. After all, this is a eukaryotic cell, which means that it does have a nucleus. And so division isn't just a simple chop in half here. It's, alright, take that nucleus, break it down, move copies of the DNA to either side, which happens in these phases here, prophase, metaphase, anaphase, telophase, which we will study more in later lectures. And then the actual cell gets broken apart in a phase called cytokinesis. So overall, the eukaryotic cell cycle, much more complex. We've got G1 for cell growth, S phase for copying that DNA, G2 for more growth and preparing for division, and then division itself, which has many subparts. All these other phases, the G1, the S phase, and the G2 together are referred to as interphase. So that's inter means between, so interphase is the phase between cell divisions, where most cells spend their time. In some cases, there are cells that will never divide again, such as in your brain. Those cells leave the cell cycle during G1 and enter a phase called G0. But beyond those exceptions, since the beginning of life on this planet, cells have completed some version of this cell cycle over and over again to get from Luca to you.